Hello friends, welcome to BMH Learning. This video is about estimation of glucose in honey by Wilstatters method. So, the aim of the experiment is to determine the glucose content in the honey sample. We are well aware that honey contains the glucose, but actually, glucose is not the only sugar present in honey, but instead it also contains sucrose and fructose. But in this method, we will only estimate the glucose content in the honey. Principle of Wilstatter method is based on the iodometric determination of glucose. Iodometry is a type of volumetric chemical analysis method and a redox titration in which either the appearance or you can say the disappearance of elementary iodine indicates the endpoint. Iodometry is widely applied in analyzing reducing compounds, as in case of glucose having aldehyde group. The aldehyde group can be easily oxidized to the carboxylic group to form gluconic acid. So iodine is actually reacting with both glucose and sodium hydroxide. By reacting with base, it forms sodium iodide. And some part of iodine oxidizes the glucose to form gluconic acid. And the remaining unreacted iodine is titrated against standard sodium thiosulfate solution in the presence of starch, which acts as an indicator. This reaction is quantitative in basic environment because the carboxylic group is acidic in nature and hence forms salts with bases. Requirements. This is the list of the chemicals which you need to carry out the experiment. You can have a look at these. Now let's proceed to the procedure. So first of all, sample of honey is added into a beaker containing hot water. The hot water will properly dissolve the honey in it. Next we need to dilute the sample. For that, first transfer the honey solution into a measuring cylinder or a flask. Make the volume to 100 ml with distilled water. From this 100 ml diluted solution of honey, take 10 ml and transfer it into another bottle. Into this bottle, add 5 ml sodium hydroxide and 25 ml iodine solution. Before the iodine is added, the solution is colorless, but addition of iodine makes it brown colored. Now, the reaction mixture is shaken to mix the contents. A plug or a lid is placed at the mouth of the bottle. This lid actually moistened with 5% potassium iodide solution. Then, keep the reaction mixture in dark for approximately 30 minutes. Once 30-minute period is over, add 5 ml of 2-normal sulfuric acid. So that means we are acidifying our sample. Now, the iodine will be liberated in the solution. Before the iodine is lost, we titrate it against sodium thiosulfate. So drop by drop, sodium thiosulfate is added. Now, when the brown color of the solution is turned into yellow by the continuous addition of sodium thiosulfate, at that exact point, we have to add the starch indicator. The addition of starch will cause the reaction mixture to turn into blue color. But at the same time, we have kept on adding sodium thiosulfate. Its continuous addition changes the blue color solution into transparent. We are gonna do this for blank also, in which instead of honey sample, we use only distilled water. And the rest of the procedure is same. The only difference is we have to add 10 ml iodine solution instead of 20 ml as used in main experiment. Next is the standard titration. For this, we have to weigh 0.245 gram of potassium dichromate. Make a solution of potassium dichromate by adding the weighed amount in distilled water and make the volume to 100 ml. From this 100 ml, take 10 ml and transfer into a conical flask. Into this, add 5 ml of both concentrated HCl and 5% potassium iodide. Now, mix the contents of the reaction mixture. Immediately titrate the reaction mixture against sodium thiosulfate solution. As the solution turns into yellow, add starch indicator, which will cause the formation of blue color. Keep on adding until the blue color turns into green. At this point, the reaction is complete. This was all. Thanks for watching.